dear students. This is the PowerPoint presentation which is explaining you about the remaining part of the ultrastructure of eukaryotic cell or the animal cell. So in the previous class we have seen up to the cell membrane or the plasma membrane, and the next important component is the cytoplasm. Next important component of the animal cell is the cytoplasm. So what this cytoplasm is? This cytoplasm is usually clear in color, that is, it is almost like a transparent color and it has a gel-like appearance. What does this cytoplasm compose of? This cytoplasm mainly consists of water, and along with water it has many enzymes. The cytoplasm has much of salt. Then it has several cell organelles. Organelles are nothing but cell organelles, which are like the endoplasmic reticulum, Golgi complex, mitochondria, ribosomes, lysosomes. All these are the cell organelles. And in addition to these three substances, that is the enzymes, salts, cell organelles, there are various organic molecules also which are present inside the cytoplasm. And you can divide the cytoplasm into endoplasm and the ectoplasm. For example, if this is the cell is entirely filled with the cytoplasm. This whole dotted region is the cytoplasm. And this cytoplasm can be divided into two parts as you see here in the third point. The first part is the endoplasm and the second part is the ectoplasm. The part of the cytoplasm which is on to the outside, just very close to the cell membrane, is called as the ectoplasm. And the part of the cytoplasm which is towards the inner side of the cell is called as it. You can observe that this cytoplasm is located very much inside of the cell. This is called as the endoplasm. So the cytoplasm which is nearer to the cell membrane is called as ectoplasm, and the cytoplasm which is located towards the inner side is called as the endoplasm. So how do they look like? Endoplasm is the central area of the cytoplasm, and usually this part contains all the organelles. And what is ectoplasm? Ectoplasm is more gel-like portion. It is more gel-like portion, and it is the peripheral portion. Peripheral is nothing but it is present very close to the cell membrane, or it is present on the outer side of the cell, outer portion of the cell, that is very near to the cytoplasm, near to the cytoplasm or plasma membrane, plasma membrane. And what this cytoplasm consists of? Cytoplasm mainly consists of three main components. One thing is the cytosol, the other one is the organelles, and many other substances which are called as cytoplasmic inclusions. We call them as cytoplasmic inclusions. And how does this cytosol look like? It is a semi-fluid or a liquid part of the cell cytoplasm. Generally, if you observe its location, it is located outside of the nucleus, but it is present within the cell membrane. It is present outside of the nucleus, that is, if this is the cell and this is the nucleus, this part is outside of the nucleus, but it is inner when you compare with the cell membrane. This is the cell membrane and this is the nucleus. So if you compare the cytoplasm position or the cytosol position, it is located outside the nucleus, but it is within the cell membrane. It is inside to the cell membrane. And then what are the organelles here? Organelles are nothing but the tiny structures which perform different functions, specific functions inside a cell. And the best examples of organelles are the mitochondria, the ribosomes, nucleus, lysosomes, chloroplasts, endoplasmic reticulum, Golgi apparatus, as well as the cytoskeleton. All these parts are termed as the organelles. All these parts, they perform different functions, and this cytoskeleton especially, it maintains the cell shape and it provides support to the organelles. Just like a skeleton, it will act as a skeleton to the cell. It will help the cell to maintain its shape and provide support for the organelles. And then what are the cytoplasmic inclusions here? Cytoplasmic inclusions are nothing but the particles which are suspended in the cytoplasm, but they are present only temporarily. These particles are present only temporarily. They are not found always. They are found only temporarily in the cytoplasm. So inclusions consist of the macromolecules as well as the granules. Macromolecules as well as the granules. And you can classify these inclusions into three types. Inclusions ni మనము త్రీ టైప్స్ కింద క్లాసిఫై చేయొచ్చు. And these inclusions, three types of inclusions are namely secretory inclusions, nutritive inclusions, and the pigment inclusions. So this is type 1, this is type 2, and this is type 3. So we have three types of inclusions. First one is secretory inclusions, second one is nutritive inclusions, third one is pigment inclusions. So examples here you can see that secretory inclusions అని దేని అంటాం. సెక్రెషన్ అంటే అర్థం ఏంటంటే స్రావకము. శ్రావకం తెలుగు వాళ్ళకి తెలుగు మీడియంలోంచి ఇంగ్లీష్ మీడియంలోకి వచ్చిన వాళ్ళ కోసం ఈ వర్డ్ వాడుతున్నాను, శ్రావకం. Secretions are nothing but the productions or something watery like substance or the substances which are produced outside the cell are called as a secretions. దీనికి బెస్ట్ ఎగ్జాంపుల్స్ యూ కెన్ కోట్ as proteins, enzymes as well as the acids. న్యూట్రిటివ్ ఇంక్లూషన్స్ ఏంటి మరి? పోషక ఇంక్లూషన్స్ అన్నమాట. న్యూట్రిటివ్ ఇంక్లూషన్స్ని గ్లైకోజన్ ఆర్ లిపిడ్స్గా చెప్పుకోవచ్చు. The lipids and the glycogen present inside the cell can be classified as the nutritive inclusions. And apart from these two, the third type is the pigment granule inclusion. The best example for this pigment granule inclusion are the melanin, which is found in the skin cells. See, you can see what are the inclusions here. These are the cell inclusions. This is the cytoplasm. This entire region is the cytoplasm. You can see this light blue color part here. All that is the cytosol or the cytoplasm part. And you can see the different organelles, green colored, brown colored, red colored structures, which are nothing but the ER, and this is the Golgi complex, this is the mitochondria. All these are termed to be the organelles. But if you observe here, there are small dot-like structures, right? These small dot-like structures are referred to the cell inclusions. These cell inclusions are namely three types. These three types we have seen here. These three types we have seen here. And then the next part, the next component after the cytoplasm, the most important part inside the cell are the cytoplasmic organelles. Let us look at each cell organelle one by one. The first important cell organelle is the endoplasmic reticulum. Antarjeeva Dravyajalakamu for Telugu medium students. Endoplasmic reticulum, Antarjeeva Dravyajalakamu. What this membrane is? This membrane is a continuous membrane system. ఎక్కడ కట్ అయిపోకుండా కంటిన్యూస్గా ఉండేటువంటి ఒక వ్యవస్థ ఇది. దెన్ హౌ ఇస్ ఇట్ ఫార్మ్డ్? దిస్ ఎండోప్లాస్మిక్ రెటిక్యులమ్ ఇస్ ఫార్మ్ విత్ ద హెల్ప్ ఆఫ్ ఫ్లాటెండ్ సాక్ లైక్ స్ట్రక్చర్స్. అంటే ఒక సంచి తీసుకొని దాన్ని అణిచేస్తే ఎలా ఉంటుందో అలాగా ఉన్నట్లుగా కనిపిస్తుంది మనకి. ఇట్ కన్సిస్ట్ ఆఫ్ అ సిరీస్ ఆఫ్ ఫ్లాటెండ్ సాక్ లైక్ స్ట్రక్చర్స్ విచ్ ఇస్ ప్రెసెంట్ విత్ ఇన్ ద సైటోప్లాజమ్. అండ్ వాట్ ఈస్ ఇట్ డూయింగ్? వాట్ ఈస్ ఇట్స్ ఫంక్షన్? ఎండోప్లాస్మిక్ రెటిక్యులమ్ ఎందుకు ఉండాలి సెల్లోని? Because this endoplasmic reticulum performs the function of synthesis, performs the function of protein folding, lipids, as well as performs the function of modification of proteins and the transportation of the proteins. So these four are the functions of the endoplasmic reticulum. It synthesizes proteins, folds the proteins, modifies the proteins, and then transports the proteins. Along with the proteins, it also synthesizes small lipids also. Then let us look at a history of this. Keith Potter, Albert Claude, Ernst Fullman. These three are the scientists who have described this structure. They said that this structure is highly convoluted. Convoluted is nothing but folded structure. Highly convoluted structure, and it looks like a brain anamata. It looks like brain. It is called as labyrinthine structure of the endoplasmic reticulum. And then Porter, Helen Thompson, Francis Kallman, these three scientists, they have for the first time introduced the term endoplasmic reticulum. And Porter and Pallet, later they gave all the important properties of the endoplasmic reticulum. So if you see the endoplasmic reticulum, there are three series of scientists, Porter, Claude, and Fullman, who have described that endoplasmic reticulum is highly convoluted. Second group of scientists, that is Porter, Thompson, and Kallman, they have introduced the term endoplasmic reticulum. third group of scientists, Porter and Pallet, what they have done is they have in detail given us an idea about the characteristics of ER and the endoplasmic reticulum taluka lakshana. See, these are all the different parts of endoplasmic reticulum. You can see the nucleus here, and attached to this nucleus you can see some sac-like structures. Here you can see the sac-like structures. These are all the sac-like structures. All these sac-like structures are nothing but the endoplasmic reticulum. But you can see that there are two sac-like structures here. One is rough endoplasmic reticulum, the other one is smooth endoplasmic reticulum. You can observe that this part is called as rough endoplasmic reticulum because it is having blue dot-like structures, and these blue dots are nothing but the ribosomes. So because this endoplasmic reticulum is having a ribosome on its surface, it is called as RER or rough endoplasmic reticulum. And if you look at this part of the cytoplasm, endoplasmic reticulum, which is light blue in color, this is not having any blue dots. No blue dots are present. That is, ribosomes are not present. And this part of the endoplasmic reticulum is called as SER, smooth endoplasmic reticulum. And this smooth endoplasmic reticulum is involved in lipid synthesis. This rough endoplasmic reticulum is involved in protein synthesis. So this is how you describe the endoplasmic reticulum. And then let us look at the rough endoplasmic reticulum exclusively here. This is the diagram showing you the cell, entire internal structure of the cell, and in this cell, only this highlighted yellow color part is showing you only the rough endoplasmic reticulum. It is showing you only the rough endoplasmic reticulum. This part has been separated and it is being enlarged in this lower diagram to show you what is there on the surface. You can see that the rough endoplasmic reticulum is covered with ribosomes, and ribosomes are nothing but protein making machines. రైబోజోమ్స్లోనే ప్రోటీన్స్ అన్ని తయారవుతాయి కాబట్టి ప్రోటీన్లను తయారు చేసేటువంటి యంత్రాలు అని చెప్తాం. Proteins making machines are nothing but the ribosomes. So what will happen in this rough endoplasmic reticulum? Rough endoplasmic reticulum will always modify the proteins that should be transported to other parts of the membrane, like surface of the cell or outside of the cell. So it will be modifying the proteins. The three main functions of endoplasmic reticulum are: it synthesizes proteins, it folds the proteins after synthesis, after folding it sorts the proteins. Sorts ante వేరు చేయటం అన్నమాట, వేరుపరచడం అన్నమాట. Okay, the same explanation I have given in the form of points here. So the rough endoplasmic reticulum, it sends signals in case the protein folding goes wrong, then protein sorting will not take place. This endoplasmic reticulum immediately sends signals to the nucleus that there is some problem in the protein synthesis and folding has not properly occurred. So protein translation immediately stops. Thereby, no faulty protein or no defective protein is synthesized. Okay, next, the smooth endoplasmic reticulum. Now you can concentrate only on this part of the endoplasmic reticulum, which is smooth, which is not having any ribosome. See, you can see that this endoplasmic reticulum is smooth because it has no ribosomes on the surface. So it will not be performing the functions of protein synthesis. But then what will it do? Why is SER there? Yes, smooth endoplasmic reticulum is there because it synthesizes the lipids such as fatty acids, it synthesizes the phospholipids, it synthesizes the steroids. And second important function of the smooth endoplasmic reticulum is it detoxifies the molecules. Detoxification అంటే అర్థం ఏంటంటే నిర్విషీకరణ చేయటం, విష లక్షణాన్ని పోగొట్టడం, టాక్సిక్ ఎఫెక్ట్ని పోగొట్టడాన్ని లేదా న్యూట్రలైజ్ చేయడాన్ని we call it as a detoxifying molecule. So SER or the smooth endoplasmic reticulum helps to detoxify the molecules like alcohols, drugs, and then other metabolic waste products. మన బాడీలో చాలా మెటబాలిక్ రియాక్షన్స్ అవుతూ ఉంటాయి. మెటబాలిక్ రియాక్షన్స్ అయ్యేటప్పుడు వేస్ట్ ప్రొడక్ట్స్ ఫామ్ అవుతూ ఉంటాయి. సో టు ఎలిమినేట్ దిస్ వేస్ట్ ప్రొడక్ట్స్ ఆర్ టు న్యూట్రలైజ్ ద ఎఫెక్ట్ of these waste products, the smooth endoplasmic reticulum comes to play a very important role. And then other important functions are it helps in the metabolism of carbohydrates. Similarly, endoplasmic reticulum in case of muscular system and in case of nervous system, endoplasmic reticulum has different functions. In case of muscular and nervous system, endoplasmic reticulum plays a different function. It will store and release the calcium ion for the functioning of this nervous and the muscular system. So it has additional function in the nervous as well as in the muscular system, right? So here let us look at the points once. Endoplasmic reticulum, SER, does not have ribosomes, so it is involved in the synthesis of the lipids, including the cholesterol, phospholipids, and steroids, plays an important role in the steroid hormone synthesis, helps in detoxification of drugs and other harmful substances in the liver cells. Ante liver cells lo ఉన్న smooth endoplasmic reticulum, it will take up the function of this detoxification of drugs as well as the harmful chemicals. Along with that, we have muscle cells and nerve cells also. In the muscle cell and nerve cell, this same smooth endoplasmic reticulum, it regulates the calcium ion concentration, helping the muscle to contract as well as to relax. It will regulate the calcium ion concentration, helps the muscle to relax or to contract, or to contract. So we have seen general endoplasmic reticulum, the rough endoplasmic reticulum, as well as the smooth endoplasmic reticulum. Then the next important part is the Golgi apparatus, or which is also called as Golgi complex. Next important cell organelle is the Golgi apparatus or Golgi complex, and it was observed in 1897 by an Italian cytologist named Camillo Golgi. He was working on some what we call owls, and he came to observe this Golgi complex, and he found that this Golgi complex also has a membrane, and it is also made up of flattened sac-like structures which are called as cisternae. So the Golgi complex is made up of flattened sac-like stacked pouches like this. It looks like this. Golgi complex looks like this. Stack of pouches, flattened pouches. These all are called as the cisternae. These are called as cisternae. So what does it do here? This Golgi complex, it will help in the transporting, modifying, packaging of proteins and lipids into small vesicles and sends them to the targeted destinations. What is the targeted destination mean? That is, some of the proteins must reach the cell membrane, some of the proteins must go to the cytoplasm, some of the proteins must go to the lysosomes, and some of the proteins must go to the peroxisomes. Like they have to go to different organelles. So who will tell them to go? See, you are a you are an enzyme of lysosome. You have to go to the lysosome. Who is going to tell? No one is going to tell. It is the Golgi complex which will take the molecule and deposit in the cell membrane. Golgi complex takes the molecule, deposits in the cell membrane, cytoplasm. Golgi complex takes the proteins and gives it to the lysosomes. Golgi complex takes the proteins and gives them to the peroxisomes. So it will be helping in transporting the proteins, modifying the proteins, packing the proteins and lipids which are synthesized by the endoplasmic reticulum. ఎండోప్లాస్మిక్ రెటికులం సింథసైజ్ చేసిన తర్వాత వాటిని తీసుకెళ్లి సెల్ ఆర్గనల్స్కి, వేరే సెల్ ఆర్గనల్స్కి ఇచ్చేటువంటి ఫంక్షన్ గాల్జీ కాంప్లెక్స్ది. And if you look at the position of the Golgi complex, ఎక్కడ ఉంటుంది Golgi complex? This is located in the cytoplasm. సైటోప్లాజంలో ఎక్కడ ఉంటుంది? It is next to the endoplasmic reticulum. ఎండోప్లాస్మిక్ రెటికులం పక్కనే ఉంటుంది, అలాగే సెల్ న్యూక్లియస్ పక్కనే ఉంటుంది, కేంద్రకం పక్కనే ఉంటుంది, right? And if you observe the plant cells, because they have so many secretions, several Golgi complex are formed. అంటే secretions ఎక్కడైతే ఎక్కువ ఉంటాయో Golgi complex number will be more in number, right? Generally a Golgi complex is made up of 4 to 8 cisternae, right? But sometimes there are some single celled organisms where there are about 60 cisternae, right? And then these cisternae, that is the stack like structures, 1 2 3 4, ఇలాంటివి 8 ఉంటాయి, right? These structures are internally, they are what we call associated or bound together by proteins which are called as the matrix proteins, right? These are small tube-like structures, microtubule-like structures. And if you look at these cisternae, ఈ సిస్టర్నేలోని మూడు కంపార్ట్మెంట్స్ ఉంటాయి. ఒకటి సిస్ కంపార్ట్మెంట్, ద సెకండ్ వన్ ఇస్ ద మీడియల్ కంపార్ట్మెంట్, థర్డ్ వన్ ఇస్ ద ట్రాన్స్ కంపార్ట్మెంట్. సిస్ కంపార్ట్మెంట్ దగ్గరగా ఉంటుంది దేనికంటే ఎండోప్లాస్మిక్ రెటికులంకి. మీడియల్ కంపార్ట్మెంట్ మధ్యలో ఉన్నటువంటి సిస్టర్నేని మీడియల్ కంపార్ట్మెంట్ అంటాం. Trans compartment, it is far from the endoplasmic reticulum. Just let me show you the diagram here, right? Just look at this diagram here. This is the transmission electron microscope diagram, which is in black and white, and this is the color picture. Okay, now if you are seeing this, this is the cis phase of the Golgi complex, and this is the trans phase of the Golgi complex, and this is the median part of the Golgi complex. Cis phase మనకి ఇక్కడంతా ఎండోప్లాస్మిక్ రెటికులం ఉంటుంది. ఎండోప్లాస్మిక్ రెటికులం దగ్గరగా ఉంటుంది. ట్రాన్స్ ఫేజ్ మనకి away from the nucleus. Away from the nucleus. See, you can see that it is receiving the proteins in the form of vesicles from the endoplasmic reticulum. Okay, it is making it is sorting inside these vesicles. It is again releasing them out to the destinations. ఇక్కడ లోపల మళ్ళీ modifications జరిగి ఆ proteins destinations కి reach అవుతాయి. You can also see here, this is the cis side where you can see small vesicles, small cisternae, and this is the trans side which we can see only the vesicles are there. In the medium part you can see sac like structures. This is the median part. So మూడు భాగాలు అన్నమాట. cis part, trans part, మధ్యలోనేమో medial part, right? So what is the cis phase and trans phase doing? Cis phase is also called as the receiving phase of the Golgi apparatus because it receives all the vesicles which are coming from the endoplasmic reticulum. And if you observe, the cis phase is convex in shape. It looks like this, convex in shape, right? So then it also faces the endoplasmic reticulum. It receives the vesicles from endoplasmic reticulum. It receives all unmodified proteins. So all the proteins which are coming from the endoplasmic reticulum are unmodified proteins. అంటే ఇంకా modifications జరగాలి. Golgi complex లోని modifications జరుగుతాయి. So the trans phase చూస్తే it is the exiting phase, not existing phase. It is the exiting phase, and the phase which is leaving the Golgi. Here the vesicles leave the Golgi apparatus. You can see here the vesicle is coming out of the Golgi complex. The vesicle is leaving. Here the vesicle is leaving. The vesicle is leaving. It is coming out. It is usually concave in shape. You can see that ఇది కాన్వెక్స్ అయినప్పుడు ఈ లోపల సర్ఫేస్ ని మనం కాన్కేవ్ అని చెప్తాం. ఇది కాన్వెక్స్. This is convex and this shape is concave. Obviously when one surface is convex, the other surface is concave, and usually it faces the cytoplasm, and then vesicles come out from the Golgi complex. Here in the Golgi complex, proteins are modified and they leave the Golgi complex. అంటే ఇక్కడ మీకు వస్తున్నటువంటి సిస్టర్నేలో let me erase the ink to show you clearly. Here see, you can see these proteins are modified proteins. These are the modified proteins. Okay, these are the modified proteins. These two, these are also the modified proteins. But whereas these proteins are the unmodified proteins which are coming from the ER. Unmodified proteins. Unmodified proteins from ER. So this is the difference. Modifications takes place in these channels or the cisternae. Then the modified protein comes out and goes to the cytoplasm or goes to the cell membrane, goes to the lysosomes, goes to the mitochondria, goes to the peroxisomes, several other cell organelles which are present inside the cell. Then see, you can see the modifications, how the modifications takes place here. See, stepwise. This is 1, 2, 3, 4, then 5, and then 6. You can see how the protein is modified. In the step 1, vesicles move from endoplasmic reticulum to Golgi. This vesicle is slowly coming. It is fixing on this Golgi complex. In the second step, you can see that vesicles coalesce, that is join. వెసికల్ కోయలైజ్ అవ్వటం అంటే జాయిన్ అవ్వటం టు ఫార్మ్ ద న్యూ గోల్జీ సిస్టర్ని. అగైన్ దిస్ విల్ ఫార్మ్ అనదర్ సిస్టర్ని హియర్, అనదర్ సిస్టర్ని హియర్ లైక్ దిస్. ఇన్ ద థర్డ్ స్టెప్ సిస్టర్నల్ మ్యాచురేషన్ అంటాం దీన్ని. ఇక్కడ Golgi sister name move అవుతూ ఉంటుంది. As the sister name move, they see here, as the proteins move, there are so many enzymes in the sister name, and all these proteins they undergo interactions with these enzymes and they get modified. Then fourth step, vesicles form and leave the Golgi. After modification, you can see that a small cup is coming from this side. A small cup is coming from this side with the modified proteins. This phase is called as the trans phase. And in the fifth step, what is happening is vesicles will transport some of the proteins to the Golgi itself, because they are needed by the Golgi also, or some of them are transported to the endoplasmic reticulum for the transport back to several other organs. This is the modification of biomolecules which is taking place in the Golgi complex. And then the next important cell organ. So till now we have seen endoplasmic reticulum, we have seen the Golgi complex. The third important structure is the ribosome. The third important organelle is the ribosome. We have seen that ribosome is called as the protein making machine of the cell. Protein తయారు చేసే యంత్రం. See, you can see that. How do you find these ribosomes? Ribosomes, we can find them as free particles in the cytoplasm, or they may be attached to the endoplasmic reticulum also in case of eukaryotic cells. And these were first described by American cell biologist who is called as George E. Pallet. Then if you look at the size of the ribosome, size of the ribosome depends on the type of the cell. Okay, now it also depends on whether the cell is resting or it is dividing or replicating, depending upon the, అంటే మనకి ఎంత ప్రోటీన్ కావాలో ఒక సెల్కి, ప్రోటీన్ రిక్వైర్మెంట్ ఎక్కువ ఉంటే నంబర్ ఆఫ్ రైబోసోమ్స్ మోర్ ఉంటాయి, ప్రోటీన్ రిక్వైర్మెంట్ తక్కువ ఉంటే నంబర్ ఆఫ్ రైబోసోమ్స్ ఇన్ దట్ సెల్ విల్ బి లెసర్ ఇన్ నంబర్, రైట్? And then this ribosome is made up of proteins which are called as ribosomal proteins and ribosomal RNA. So ribosome is made up of ribosomal, what is ribosome made up of? Ribosomal proteins. రైబోజోమ్ నిండా రైబోజోమల్ ప్రోటీన్స్ ఉంటాయి, అలాగే రైబోజోమల్ ఆర్ఎన్ఏ ఉంటుంది, రైట్? అండ్ ఈచ్ రైబోజోమ్ ఇట్ హ్యాస్ టూ సబ్ యూనిట్స్. యూ కెన్ సీ హియర్, దిస్ ఈస్ ద లార్జర్ సబ్ యూనిట్ అండ్ దిస్ ఈస్ ద స్మాలర్ సబ్ యూనిట్. ఉప ప్రమాణాలు అంటాం తెలుగులో అయితే. So every ribosome is made up of two subunits, larger subunit and a smaller subunit. And these subunits are named as S. ఇక్కడ చూస్తే మనం దీన్ని, if it is a eukaryotic ribosome, it is called as 60S ribosome and this is called as 40S ribosome. ఇలా S అని ఎందుకు అంటామంటే అది స్వెడ్బర్గ్ యూనిట్స్ అనమాట. సో దాన్ని సెడిమెంటేషన్ అంటే అవక్షేపం కింద, లేదా ఏమంటారు, అవక్షేపం కూడా కాదు, సెడిమెంట్ లేదా స్ఫటిక రూపంలో ఫామ్ అయిపోయేదాన్ని సెడిమెంటేషన్ రేట్ అంటాం మనం. ఆ స్వెడ్బర్గ్ యూనిట్స్కి మనం ఇక్కడ ఎస్ అని పెడతాం. సెంట్రిఫ్యూజ్ మిషన్లో వేసి తిప్పినప్పుడు ఈ యూనిట్స్ సెడిమెంటేషన్ అయ్యేటువంటి రేట్ ఏదైతే ఉందో, ఎంత వాల్యూ దగ్గర అయితే సెడిమెంట్ అవుతుందో, అవక్షేపంలాగా మీకు ఏర్పడిపోతుందో, దాన్ని మనం స్వెడ్బర్గ్ యూనిట్ల కింద చెప్తాం. And the large and the smaller subunits of eukaryotic cell are the 60S and the 40S. Smaller subunit is the larger subunit. Let us look at the function of the ribosome. What will it do? See, ribosome ni open chest chuste, these are the sites you find inside the ribosome. This diagram is very important. Ribosome is the place where the information which is carried in the form of genetic code is converted into protein molecule. mRNA దగ్గర నుంచి ఇన్ఫర్మేషన్ తెచ్చుకుని it will translate the information which is present on the mRNA into the protein. ఇది రైబోజోమ్ చేసేటువంటి పని. ఓకే, now ribosome, these ribosomal molecules of mRNA, they this just leave off the second point because it is a little bit complicated. To make it much more easier, we will see the fourth point here. See how this different molecules come. అంటే ఇక్కడ మనకి డిఫికల్టీ అంటే మనం అర్థం చేసుకునే దాన్ని బట్టి ఉంటుంది. ఇఫ్ యూ అబ్జర్వ్ మీకు ఈ పదాలు తెలిసినవే అవుతాయి. ఎంఆర్ఎన్ఏ ఒకటి, టీఆర్ఎన్ఏ ఒకటి వినే ఉంటారు. ఆర్ఎన్ఏ 3 టైప్స్ ఆఫ్ ఆర్ఎన్ఏ అంటాం. బాటనీలో ఎక్కడ వచ్చి ఉంటాయి. ఎంఆర్ఎన్ఏ 3 టైప్స్ ఆఫ్ ఆర్ఎన్ఏ, టీఆర్ఎన్ఏ ట్రాన్స్ఫర్ ఆర్ఎన్ఏ అంటాం, అలాగే రైబోజోమల్ ఆర్ఎన్ఏ. దీస్ ఆర్ 3 టైప్స్ ఆఫ్ ఆర్ఎన్ఏస్. సో ఇక్కడ టీఆర్ఎన్ఏ ఏం చేస్తుందంటే మూడు మూడు ఉన్నటువంటి కోడ్ ఒకటి తీసుకొస్తూ ఉంటుంది. అమైనో యాసిడ్ సీక్వెన్స్ ప్రోటీన్ని మనకి తెలియజెప్తుంది. అమైనో యాసిడ్ సీక్వెన్స్ ఆఫ్ అ ప్రోటీన్ ఈస్ డిటర్మైన్డ్ బై ద ఆర్డర్ ఆఫ్ ద టీఎన్ఏ మాలిక్యూల్. ఒక్కొక్క అమైనో యాసిడ్ని తీసుకొస్తూ ఉంటుంది టీఆర్ఎన్ఏ మాలిక్యూల్. అందుకనే ట్రాన్స్ఫర్ ఆర్ఎన్ఏ అన్నాం. ఇంకా when you learn about the ribosome function in detail, then you will be understand what the tRNA molecule is doing here. And then molecules of rRNA, what will they do is they form the peptide bonds. They form the peptide bonds. If these two are the amino acids, a small peptide bond is formed. Another small peptide bond is formed, another amino acid comes. Another peptide bond is formed, another amino acid comes. So this function of peptide bond formation is done by the molecules of RNA. And these amino acids are brought by the tRNA. tRNA brings this amino acid molecules. tRNA brings these amino acid molecules. Okay. So now you can see that there are 3 sites on this ribosome. Let me clear the ink first. See, if you look at the structure of the ribosome, inner side choose the aquila. The ribosome, every ribosome has 3 sites. This is called as amino acid site, at which the amino acid comes and a tRNA అనుకున్నాం కదా, ఒక టీఆర్ఎన్ఏ ఒక అమైనో యాసిడ్ మాలిక్యూల్ తీసుకొచ్చి ఈ సైట్లో పెడుతుంది. ఇమీడియట్గా ఇక్కడ మనకి ఆల్రెడీ ఒక టీఆర్ఎన్ఏ కొంత అమైనో యాసిడ్ మాలిక్యూల్స్ కొన్ని జాయిన్ అయిపోయి ఉన్నాయి, రైట్? సో నౌ దిస్ అమైనో యాసిడ్ ఆల్సో జాయిన్స్ టుగెదర్, రిజల్ట్స్ ఇన్ ద ఫార్మేషన్ ఆఫ్ ఎ పెప్టైడ్ బాండ్. దిస్ టీఆర్ఎన్ఏ ఇలా ఇక్కడ లీవ్ అయిపోతుంది. ఈ పెప్టైడ్ బాండ్ ఈ అమైనో యాసిడ్ ఇక్కడ రిలీజ్ అయిపోతుంది. రిలీజ్ అయిపోయిన తర్వాత లాస్ట్లో అన్ని అమైనో యాసిడ్స్, ఇంకా ఇక్కడ మనకి ఇంకొక అమైనో యాసిడ్, ఇంకొక అమైనో యాసిడ్, అనదర్ అమైనో యాసిడ్. ఆఫ్టర్ జాయినింగ్ సో మెనీ అమైనో యాసిడ్స్, this ribosome, it keeps on moving slowly on this mRNA molecule. This is the place actually where mRNA binding site is there. mRNA comes and binds on this site, and the ribosome keeps on moving on this mRNA. It reads the code. mRNA మీద ఏముంటాయి మనకి? If this is the mRNA molecule, nitrogen bases ఉంటాయి. A, T, see I'll write here, A, T, C, C, A, U, G. These are the sequences. So as the ribosome moves, it will be reading these sequences. మూడింటిని చదువుతూ ఉంటుంది. 3 చదవగానే ఒక అమైనో యాసిడ్ వస్తుంది. మళ్ళా 3 చదవగానే ఇంకొక అమైనో యాసిడ్ వస్తుంది, పెప్టైడ్ బాండ్ ఫామ్ అవుతుంది. మళ్ళా 3 చదవగానే ఇంకొక అమైనో యాసిడ్ వస్తుంది, పెప్టైడ్ బాండ్ ఫామ్ అవుతుంది. So in this way, the ribosome has special sites for the synthesis of ribosome. Imagine if a ribosome is there without these sites. So ఫంక్షన్ చేయాలంటే రైబోజోమ్కి ఈ సైట్స్ అన్నీ కావాలి. ఏ సైట్ ఒకటి, ఏ సైట్ లేకపోతే అమైనో యాసిడ్ కెనాట్ కమ్. పెప్టిడైల్ సైట్ ఆర్ పీ సైట్ మస్ట్ బి దేర్. ఈ సైట్ లేకపోతే పెప్టైడ్ బాండ్ ఫార్మేషన్ అవ్వదు. ఈ సైట్, ఎగ్జిట్ సైట్, పాలీపెప్టైడ్ చైన్ మొత్తం సింథసైజ్ అయిన తర్వాత దట్ పాలీపెప్టైడ్ చైన్ కమ్స్ అవుట్ ఆఫ్ దిస్ సైట్. సో ఇఫ్ దిస్ సైట్ ఈస్ నాట్ దేర్, పాలీపెప్టైడ్ చైన్ విల్ నాట్ బి ఏబుల్ టు కమ్. ఇదంతా బాగానే ఉంది. ఇవన్నీ ఎప్పుడవుతాయి? ఇక్కడ ఎంఆర్ఎన్ఏ బైండింగ్ సైట్ లేకపోతే there is no mRNA binding. Here you can see in this diagram. See this 5 prime, 3 prime mRNA molecule should bind at this place here, right? After it binds, the ribosome keeps on reading, moving, and you can see a purple color chain that is coming outside. This is nothing but the polypeptide chain. So ఈ 4 ప్లేసెస్లో ఏది మిస్ అయినా కూడా మనకి ribosome will not be able to perform the function of protein synthesis.